Hello, everyone. Welcome back to Inside Takara Series. My name is Kiran, and I'm Senior Manager of Marketing and Product Strategy from DSS Takara Bio India. And I'm very happy and delighted to welcome all of you to joining this session today. As you know, Inside Takara Series is a knowledge sharing initiative where we try to bring experts to discuss about different genomics technologies, different scientific challenges, and how we can overcome these challenges. So today also we have planned to talk about one advanced genomic technology, long-read RNA sequencing, and different pain points and challenges researchers faces, and how we can address or how we can overcome these challenges with the appropriate solution. So before we go into the topic, I would really like to take this opportunity to welcome our guest speaker, Dr. Ning. Dr. Ning is an associate product manager from Takara Bio USA, responsible for SMART-Seq mRNA-seq portfolio, including single-cell, bulk, high throughput, and all emerging RNA-seq application. She has received her PhD in biomedical engineering from University of California, Irvine, and has completed her postdoc in Stanford University. Her broad expertise in biomedical research, biotechnology applications, and product commercializations allow her to translate innovative genomics technologies into a solution which can address researchers' needs. She's really fascinated about advancing RNA-seq technologies to help researchers to unlock deeper biological insights using NGS. So let's welcome Dr. Ning. Hi, Ning.
Thank you. Thank you for the introduction. Hi, Kira.
Hi. Okay, so all right, let's start. So we can share our screen now, coming to our main topic. Great, thank you for sharing. So you all know that RNA sequencing has evolved tremendously over the past decade. And short read technologies has transformed transcriptomics. While today's researchers are now moving beyond gene expressions, and they are now trying to understand the diversity of transcript at complete level. They want to understand all isoforms, alternative splicing, gene fusions events. There the significant impact of long read sequencing comes. But if you see, it's not that as straightforward. It comes with certain challenges and pain points. So today, you see, we will first talk about those challenges like sample challenges, especially when we are dealing with low input RNA amount. Then library prep challenges, like when we talk about how to get full and transcript information, the reproducibility of the workflow, or how to multiplex different large number of samples together. Then some biological challenges. Are we able to detect rare and low abundance transcript? And of course, we do understand downstream analysis is again another challenge. So coming to the first challenge, which is our low input like concentration. So if you see the workflow, current workflow, what we have, they generally require the concentrations amount between 300 nanogram to 1 microgram of total RNA. But every time we don't get the projects and samples having these amount of RNA, especially when we are working with the challenging samples like single cells, which might contain certain picogram amount of RNA. Organoids, which are miniature 3D model of human organs, which are very invaluable tool for drug modeling, disease tracking, tracking the progress of the disease. There's so many things which we can do from organoid, but low input concentrations. Clinical samples are always in scarce amount. Rare cell population, which are like, for example, you are working with circulatory tumor cells, specialized immune cells, and they are very small, present in a very small number. How to get a good amount of RNA from these, or how to make sure that we are going to capture these rare cell transcript or information. Cell-free RNA, we do know that it's absolutely sometimes coming fragmented, and it's present in a very low concentration. So these are the challenges when we talk about the low input. Coming towards our second challenge, library prep. So how to capture the true full length transcript, how to make sure we are getting reproducible results, and how to increase our multiplexing capability. So if you see, what is the a whole agenda of doing long read RNA sequencing? Because we want to capture the entire length of the RNA molecule from start to end, without even cutting them and then try to fix them, right? So what if we are not able to get complete cDNA or incomplete cDNA? What it will create, we will not get the complete transcript information, right? And when we are not able to get full length transcript information, the impact will be that we will have a reduced representation of transcriptome, which will not give you the complete information about your isoforms. So full length transcript recovery becomes really challenging. Second, workflow reproducibility. We do work with different library prep kits. We have different operators, and multiple steps are involved in library preparation. So we always see there some kind of variable or some kind of variability can enter in your library quality. We want to reduce that. We want to increase the reproducibility between the experiments. That's another challenge. Then third is, can we pool larger number of samples together, like increase the multiplexing capabilities? Because most of the time when we get high throughput projects, and if we are not able to pool large number of samples together, then sequencing cost can increase. So these are few of the library prep challenges which we are facing in long-read RNA sequencing. Now I am going to talk about this major challenge, how to detect rare and low abundance transcript. So if you see, a cell may express multiple genes, but every gene may not have higher number of copy, right? So coming to my next slide, if you see, rare transcripts are generally present in a very a low number. They will have a few RNA molecules. And when we know that there are only few RNA molecules for this rare transcript, but they are very important biological molecules. Very, very important. Like as I said, immunized cells, then stem cells, CT cells, right? But they are present in a low number. They are present in low abundance. If we do miss them during library preparation, because we were working with certain library prep kit which were not able to capture these transcripts, we didn't have the full length transcript and we missed this. So we are not able to capture these important biological molecules, which has a lot of significance. So these are few of the challenges which researchers are currently facing in long-read RNA sequencing. And the question is that how to get full-length transcriptome information, even if we have low concentration, without compromising in sensitivity, without compromising on the diversity of the transcript data. So here I would like to definitely invite now my guest speaker, Dr. Ning, who will actually help us to understand how we can overcome these challenges and what are the solutions present with us which can take care of these concerns, and if there are any case studies which can give us more knowledge on it. So I'm happy to invite Dr. Ning. So yeah, Dr. Ning, over to you. Please start discussing and letting us know the insightful knowledge, how we can overcome these solutions.
Oh, hi, Karen. Thank you very much for the introduction, and also the introduction for the challenges currently the long-read workflow is facing. And thank you, everyone, for joining us today for the session. And my name is Ning, and we're from Takara Bio USA, and responsible for the SMART-Seq mRNA sequencing portfolio. As Kieran discussed, long-read RNA sequencing offers tremendous potential for understanding transcriptome complexity. But several technology challenges still remain, and particularly when working with low input material and trying to recover the complete transcripts. So today I will discuss why the full-length transcript information matters, introduce the SMART-Seq mRNA long-read workflow, review its performance, and also share our recent application study demonstrating the types of isoform level biology that long-read sequencing can review. And hopefully from my presentation, you can understand how we can use this technology to conquer all the challenges Kieran mentioned earlier for long-read. So first, before I go to discussing the workflow, I would like to first to explain why long-read mRNA sequencing has become an important tool in transcriptomics. So its key advantages is the ability to directly sequence full-length transcripts. This enables researchers to identify the completed transcript structures and distinguish among different isoforms produced from the same gene. So with short-read RNA sequencing, transcripts are fragmented and reconstructed computationally. This approach is highly effective for gene expression analysis, but it can be difficult to determine which exons belongs to the same transcript, particularly for a gene with complex splicing patterns. Long-read sequencing provides more direct information about a transcript structure, supporting applications such as novel isoform discovery, alternative splicing analysis, fusion transcript detection, and transcript level studies of disease biology. So from the the second part, as you can see that this is the study from the gene code, and so in 2024. And as you can see, it added tens of thousands of newly annotated genes and more than 140,000 additional transcripts, many of which were identified through long-read sequencing technologies. These discoveries demonstrate that the transcriptome complexity is far greater than we previously appreciated, and that many biologically relevant transcripts remain undetected using earlier approaches. And then the third application will be tissue development, and also the disease research is a key application. And one thing I want to mention here is on top of the reasons I introduced here, applications, there is a big potential for long-read. So which we are opening a door that the short-read sequencing previously cannot reach. And talking about the disease model, this is an interesting study recently published in Nature Genetics. And the study focused on microglia, the resident immune cells of the brain, which play a central role in neurodegenerative disease such as Alzheimer's. So the researchers here used long-read RNA sequencing to build a comprehensive atlas of full-length microglia transcript isoforms and investigate how alternative splicing contributes to the disease. And from this image, you can see this figure summarizes one of the key findings from the study. So the researchers profiled the transcript isoforms expressed in microglia. Importantly, they didn't just detect the previously annotated isoforms, they also identify a large number of novel isoforms shown here in the middle in red. So these red transcripts were not present in the existing reference annotation. So the researchers generated an expanded transcript reference using the long-read sequencing data. They then integrate this new reference with genomic and short-read RNA sequencing data to perform downstream analysis. So you can see short-read and long-read are actually complement each other. And by combining these complementary data sets, they gained new insights into microglia biology, alternative splicing, and molecular mechanisms associated with Alzheimer's disease. So this study looked at the isoform expressed in microglia, and this large number of novel isoforms is very, very important as is previously not presented in the reference annotation. And from the study, the researchers then examined several Alzheimer's disease risk genes, including this TRIM2 And using the expanded transcript annotation generated by long-read sequencing, they identified this transcript which has not previously annotated before. And more importantly, it reviewed biologically meaningful transcript diversity that improves our understanding of Alzheimer's disease mechanisms. So from here, we can see that this is an excellent example of how long-read RNA sequencing can move beyond the transcript discovery to generate new biological insights that were difficult to obtain with conventional sequencing approaches. And more importantly, it's also showed how the data analysis can put the long-read data and the short-read together to do the data analysis and complement each other. So next, we have discussed why long-read sequencing is becoming increasingly important. And so, of course, most of you here has already heard long-read and understand that there are two major long-read sequencing platforms, which including Oxford Nanopore Technologies and PacBio. Each has its own strengths and is widely adopted by research community. So here is a quick introduction that Oxford Nanopore offers a flexible portfolio of instruments ranging from portable device to high throughput systems, making long-read sequencing accessible for a wide range of applications. And very importantly, that because it's more accessible CAPEX with more instruments placed, you can actually see this instrument not only in the big centered NGS cores, it also like you can see it in a lot of different labs. And so I hope some of you have access to this ONT sequencing as well, like for your research. And on the right-hand side, this is PacBio. It's well known for its highly accurate sequencing, making it an excellent choice for applications that require very high sequencing accuracy. So the good news is that the technology I'm going to introduce today, the SMART-Seq mRNA long-read, is compatible with both platforms from Oxford Nanopore and also PacBio. So researchers can prepare the full-length cDNA synthesis libraries using a single workflow and then sequence them on either Oxford Nanopore or PacBio, depending on their research goals and the preferred sequencing platforms. And recently, in last month, PacBio also recognized the SMART-Seq mRNA long-read, which is our kit, as a certified compatible library preparation solution. So this given researchers confidence that the workflow performs well on both major long-read sequencing platforms. So next, how does the SMART-Seq mRNA long-read kit work? As I mentioned in the previous slide, the SMART-Seq mRNA long-read serves for the long-read sequencing for the first step, which is a full-length cDNA synthesis. Starting from the total RNA, we use Takara's total RNA or single cell. We use Takara's well-established SMART technology. So just to correct, we have the input for both the total RNA and the direct cell input, which can help you save a lot of effort while preparing the cDNA synthesis. And this technology can help to generate a high quality and a full-length cDNA. So this is similar to our well-known SMART technology. The workflow begins with oligo DT primed reverse transcription and during the for the cDNA synthesis, and the workflow supports the RNA inputs are ranging from the single cell to standard bulk RNA samples, making it suitable for a wide variety of the transcriptomics applications. And once the full-length cDNA has been generated, it can be processed using the standard library prep workflow for either Oxford Nanopore or PacBio sequencing. So which is the second and the third step I'm showing here. It depends on what sequencer you are choosing. you will work with your like a downstream like a sequencing process as well. And so here is the introduction for the SMART-Seq mRNA long-read, which enables the long-read sequencing from low inputs. As Kira mentioned earlier, for the current commercial long-read kit on the market, so basically you can only see the input requirement above 300 nanogram. And with our kit, you can actually see it support the range from 10 picogram to 100 nanogram total RNA, or 1 to 1,000 cells without RNA extraction. So this way, it can help you to really enable the capability to support the direct cell input in plate-based format, or the rare sample, as Kira mentioned earlier, from the organoids to cell-free RNA, etc. So the flexibility is especially valuable when you're working with the precious or limited samples. And the second is the SMART technology is designed to generate the high-quality full-length cDNA and known for that. So we consistently capture the completed transcripts, including the transcripts longer than 8 kilobases, and enabling the accurate isoform identification and alternative splicing analysis. So in the next couple slides, I will show you some real sample, and hopefully you can appreciate the idea for reliability detection of it, so to highlight the reproducibility and also the very, very long transcripts we can deliver for the full-length transcripts as well. And the third is about a cDNA barcoding and a multiplexing. As Kira mentioned before, one challenge for long-read is once you are finishing the discovery mode, you want to have the higher throughput ability for your experiment. And also a lot of the kits are actually limited to less than 24 samples or below. But for this kit, you can actually have 8 to 96 sample and pool for sequencing, which can provide the flexibility while reducing the hands-on time. And together with this capability across with both Oxford Nanopore and the PacBio sequencing platforms, the SMART-Seq mRNA long-read can provide a streamlined workflow for generating high-quality full-length transcript data. And so now is the the real data. So starting from the single cell data, we can see the performance data for the single K56 to K562 cell direct input. So one of the biggest challenging long-read RNA sequencing is to generate a high-quality full-length cDNA from extremely small amounts of starting material. To evaluate this, we compare the SMART-Seq mRNA long-read sequencing with a leading competitor. And the first result on the left shows the read length distribution. As you can see that the SMART-Seq mRNA long-read in blue produces substantially longer reads on average, indicating more efficient recovery of the full-length transcripts. So on the right-hand side, we also look at the transcript completeness using the this ACTB genes as an example. So with the SMART-Seq mRNA long-read, you can see that 73% of read span, the full-length transcript compared with 22% of the competitor's workflow. This results demonstrate that the SMART-Seq chemistry efficiently capture the completed transcripts even from single cell input. So this is especially important for applications such as isoform identification and alternative splicing analysis, where preserving the full transcript structure is critical. So this is for the single cell comparison. And for the next slide, I'm actually showing the bulk RNA-seq data we have for this long read. And for this study, the key question is, does SMART-Seq mRNA long-read generate more complete transcript information than the major competitor workflow? In this comparison, the competitor's library was sequenced at approximately 6 times greater depths than the SMART-Seq mRNA long-read library. This is some background information. However, despite the substantially higher sequencing depths, only about 1% of reads longer than 4 kb were full length with the major competitor's workflow. In the example shown here, the competitor's workflow failed to recover any full-length reads for this particular transcript. So in contrast, the SMART-Seq mRNA long-read consistently generate full-length transcripts reads. So this improved the transcript completely translates into better resolution of the longer transcript isoforms, more reliable detection of alternative splicing and isoform spacing, and a greater sensitivity for identifying rare transcript variants. So this actually, usually I will hear a question following the comparison between our kit to other kit, even though the performance for this full length rates are definitely better. But one question will naturally raise is how long of a transcript can the workflow actually capture? So from here, I just want to introduce that in practice, the observed transcription length is usually determined not only by the technology, also by the biology of the sample together. So the figure shows the length of the length distribution of the annotated human reference transcripts. As you can see that approximately 95% of the human transcripts are shorter than 6 kilobases. So this information, 95% of human transcripts are shorter than 6 kilobases, is from the annotated human reference transcripts. Excuse me. So in other words, for most of the experiments, the transcript lengths you observe simply reflect the nature distribution of transcripts in the sample rather than a limitation of the chemistry. But the performance does show the difference with the same sample input. And if you really wanted to see the very, very long transcripts, here is the next example that we have the full length transcript approximately 15 kilobases in length generated using the SMART-Seq mRNA long-read sequencing. So this example shows that the workflow can successfully capture very long transcripts when they are present in the sample. So while most transcripts are naturally dominated by transcripts shorter than 6 kb, the chemistry itself is not restricted by that range. So it's fully capable of generating high-quality full-length cDNA from much longer transcripts, enabling the comprehensive characterization across a broad range of transcript size. And of course, it's not limit the potential of your research and your sample and your novel findings. So now I'd like to move from the performance data of the product itself in a real biological application. So this study was conducted in the collaboration with Dr. Shagi Sotanova's team at the University of Colorado in USA, and focused on understanding of how breast cancer cells adapt to the therapy. So the overall study design has 2 parts. The first part, of course, experimented the MDAMB 361 cancer cells were exposed to increasing concentration of the cancer therapy to generate a drug-resistant cell lyse. And the researchers then collected both the control and the resistant cells for comparison. So the total RNA was extracted and converted into the full-length cDNA using the SMART-Seq mRNA long-read kit. The samples were barcoded, pooled, and sequenced on the Oxford Nanopore platform. And the sequencing data were then generated to quantify the expression at the both gene and the transcript level, and to identify the full-length transcript isoforms. So this study allows us to ask two complementary questions: which genes and the transcripts change in abundance during the development of resistancy, and whether the resistant cells switch to different transcript isoforms. The second question is especially important because isoform changes can occur even when overall gene expression remains relatively stable. The long-read sequencing gives us the resolution to detect those changes directly. So in summary, this workflow connects a biological relevant model of the therapy resistance with both expression level and isoform level analysis. And one thing worth to mention is for this study, the Dr. Alina's team, they actually have their short read at the beginning, and so after finishing the study, they decided to move to long read to understand from isoform level what they can actually find. So for here, this slide is actually where we actually figure out how many of the transcripts are generally new and we can trust them. And to do that, our scientists run every transcript through SMART-Seq, which classifies each one against the latest gene code annotation. And so from the data here, you can actually show the different categories, which are the incomplete splice matches, the novel not in catalog group, and also the novel in catalog group. And you can see the number of each one of them. And so whenever you claim something is novel, the first question is it real or is this an artifact? So after we figure out like this different category, and this middle panel basically answers that the NNC isoform, which is novel not in catalog, with brand new splice junctions. 98% of them use the canonical splice size. 84% of them fall within annotated gene, and critically 72% are backed by short read coverage at every splice size. And in fact, that the most of the NNC isoforms are independently confirmed by our orthogonal SMART-Seq total RNA short read data. So these are not noise; they are credible new transcripts we find. So finally, on the right-hand side, we look at the structural complexity by the exon count. The novel NIC isoforms actually carry the most exons and more than the known FSM transcripts, which points to a more complex splicing arrangement arising in the resistant cells rather than simple truncations. So the takeaway here is long-read didn't just recover the known transcript. They actually surfaced thousands of validated novel isoforms, setting us up to ask which of these are actually switching with the therapy resistancy. So from here, you can actually see this novel transcripts, and also from here, you can see the difference between the resistant group and also the a regular wild type group. And here is this slide is before interpreting any biological differences. It's important to first establish that the data are actually highly reproducible, which is also one challenge Kira mentioned earlier for the study of the long read. So this heat map showed the Pearson correlation between all samples at both gene level on the left and also transcript level on the right. As you can see that the technical replicates cluster very tightly together in both analysis. and demonstrating the excellent reproducible through the workflow from the RNA prep and the library prep and the sequencing and also downstream analysis. So the key takeaway that is the SMART-Seq mRNA long-read generate high reproducible data, providing a robust foundation for downstream differentiation, differential expression, and isoform analysis. So this is the reproducibility of the data. And then the next would be the fine part for the gene fusion isoforms in the therapy-resistant cells. So far, we have focused on the transcript isoforms. Another major advantage for the long-read sequencing is its ability to identify fusion transcripts directly from full-length reads. So to do this, we analyzed the sequencing data retaining only the high confidence fusion transcripts supported by multiple spanning reads across exon boundaries. So using this approach, we identified 117 high confidence fusion transcripts. Of this, 18 of them are previously reported, which is on the bottom, the known fusions, while 99 of them are novel fusions, highlighting the ability of the long-read sequencing to uncover the previously undescribed fusion events. And so the panel on the right actually illustrates the representative examples of fusion transcript structures. Rather than simply identifying the two genes involved, long-read sequencing resolves the complete exon structure of each fusion transcript, providing much greater detail for downstream biological interpretation. So again, the key takeaway here is that long-read sequencing enables accurate detection and the structural characterization of fusion transcripts, including many previously unreported fusion events. And from here, continue the genome-wide fusion. This figure compared the fusion transcripts landscape across the control and the therapy-resistant cell lines. So each circus plot summarizes the genomic locations of the high-confidence fusion transcripts, with each arc representing a fusion event supported by at least 3 sequencing rates. And several fusion events are shared across all 3 cell lines, indicating the stable fusion transcripts that are characteristic of the cellular background. At the same time, the therapy-resistant cell line exhibit additional fusion events that are not observed in the control cells. You can see on in the middle and on the right. So while some of these events are shared between the resistant lines, others are unique to individual resistant populations. So this resistant associated fusion transcripts may reflect a genomic rearrangement or transcriptomic changes that emerge during the acquisition of the drug resistancy. Although additional functional studies are needed to determine their biological significance, they represent promising candidates for further investigation. So together, these results demonstrate that the long-read sequencing not only identified fusion transcripts with high confidence, but also enables researchers to compare fusion landscapes across biological conditions and uncover structural transcript changes associated with disease progression or therapeutic response. And in this particular case is for the oncology study, but we can actually think this will be like work for the tissue development and the neurodegenerative disease as we introduced earlier. And so for finally, I'd like to show a representative example of the isoform switching associated with this therapy resistant. So the panel on the left shows the relative abundance of the 2 major transcript isoforms. For an example, gene across the control and the therapy-resistant cell lines. And in the control cells, one isoform is predominantly expressed, where in the resistant cells, expression shifts towards the alternative isoform. So importantly, this also represents a change in transcript transcript usage rather than simply a change in the overall gene expression. And on the panels on the right illustrate the structural difference between these two isoforms. The transcript model show that they differ by the inclusion of the specific exon, and the sequence coverage below provides the direct evidence supporting the alternative splicing event. So that the long-read sequencing provides transcript-level resolution, enabling researchers to uncover regulatory changes that would be difficult or even impossible to detect using the conventional gene-level analysis alone. So this is a tool with definitely a lot of potential for your researchers as well. So from here, we actually review the long-read sequencing, why it's important to unlock the isoform level biology for different applications. And also we briefly introduced the SMART-Seq mRNA long-read technology can actually generate a high-quality barcoded full-length cDNA from either the single-cell input, direct input, or the total RNA. And which enable the low input studies for rare samples. And also the pooling can allow the high throughput analysis as well. And this technology is compatible with the two major long-read sequencer, which is ONT, Oxford Nanopore, and also the PacBio. So from the a comparison and benchmarking study, we can see that the technology can outperform all the different competing technology chemistry for the single cell and a Bach protocol. And also from the study with the University of Colorado, you can see that from this breast cancer study that what we reviewed can show that the long-read sequencing specifically about the SMART chemistry, it can enable the discovery and the quantification of the novel isoforms and also resolve the cancer relevant to the isoform biology as well. So you can have more information to understand the mechanism behind the resistance. So from here, I hope you have already reviewed the long-read sequencing and understand the novelty it can actually bring you and the potentially to bring to your technology as well. So from here, I would like to take any questions you might have, and thank you very much for listening to this talk. And this is good science.
Thank you so much, Ning, for sharing this all information and knowledge to us. It's so good to see these real case studies, examples, and very glad to see that the data is available both on ONT and PacBio. So a wonderful explanation, and I'm very sure that these information are definitely going to help our researchers. So Ning, we have seen some questions coming up in the chat box. So there is one question one of our audience has asked that do we have unique molecular identifiers, UMIs, in our kit, or we have a plan to have this?
Thank you, Kieran, and thank you. This is a great question. So the current version of the SMART-Seq mRNA long-read actually does not include a unique molecular identifier or UMIs. And however, we are actively developing a next generation version that will incorporate UMI support, while also expanding the supported input range and further improving the workflow performance. So even though our kit is outperformed some competing chemistries, but we are definitely actively still moving forward for this long-read product as well. UMIs can actually provide additional confidence for the transcript quantification by helping distinguish the original molecules from the PCR duplicates. So for some studies, this is definitely a plus. But in the meantime, we also have our current kit as well, so we can update development progress to you. Yeah.
Okay, great to know that. Okay. So I see another question in the chat box.
Mhm.
Why don't we can use our own direct RNA kit or cDNA synthesis kit?
Okay. This is a good question. Actually, we heard this from time to time as well. And these workflows are designed for different applications or like similar applications. Depends on what the audience is talking about, the direct RNA input or their cDNA synthesis kit. So ONT actually have both of them. Excuse me. So the direct RNA sequencing kit, which sequence the native RNA molecules without the RT or PCR, making it well suited for applications such as the RNA modification analysis. However, it typically requires substantially higher RNA input and is generally less suitable for low input or limited samples. So for researchers working with precious samples, such as like small numbers of cells and limited amount of RNA, and a SMART-Seq mRNA long-read is optimized for generating high-quality full-length cDNA from much lower input amounts. So I would say it's really relevant to the like a research purpose you are looking for. And so if the idea is to find isoforms as what I showed, then like the SMART-Seq technology would be really helpful and give you the like the best performance. But on the other hand, if the applications is different, then of course you would use different kit. And for the Oxford Nanopore cDNA synthesis kit on the other side, that's actually will generate a cDNA synthesis and for the library prep. So that's actually the idea would be very similar to SMART technology. And from the previous slide, the benchmarking slide, I already showed the performance. So you can see for like the low input, the data quality is definitely better. So thank you. Good question.
Thanks, Nick. There's, I think, one more question I see. Okay, how do I analyze the data? Do I need any specialized bioinformatics support? It's all about data analysis.
Yeah, this is another good question. So long-read is a relatively newer technology. Compared to short-read, it doesn't have that tons of like data analysis software out there. And so it's still like in development for sure. But Takara Bio actually consider about this, and the like internally, we spent quite some time to develop the bioinformatics support. So for the like a bioinformatic software called Cogent, it can actually provide the long-read analysis pipeline to simplify the primary data analysis, and it's completely free with this SMART-Seq technology. And the current release, I think it's in April 2026, and it includes a workflow for the demultiplexing, the rate alignment, and the gene level counting, which is good, pretty good to help the users to process the sequencing data from the SMART-Seq mRNA long-read workflow. And the development is also ongoing. So this is primary analysis, and the future release will add the support for the like UMI enabled workflow. Well, we provide a UMI, right, as well as additional long-stream analysis capabilities. And for more advanced analysis on top of what we can provide, such as the like isoform discovery, fusion transcript detection, or differential transcript usage. So the users can actually integrate the primary output with the from us to work together with the established long-read analysis tools, including like the Squantis-free I mentioned earlier, IsoQuantifier, or like other open source community supported software or script. So depending on the research objectives, but with the foundation for what Cogent from Takara can provide it to you, already you have a good start.
Great. Good to know this thing that we have some possibilities for the analysis also. That's, I think, definitely audience is going to be happy. So yeah, for now, I think this mini question I could see, and I would like to request audience, if you come across any question, you can always put in the chat box. We will be able to access and we'll get back to you with the answers.
That would be great.
Yeah.
Happy to support and to help you advance the research.
Absolutely. Yeah. So I really would like to thank, first of all, to all my audience for joining us in this episode. And then thanks to you, Dr. Ning, for giving us such a knowledgeful session and giving us information about SMART-Seq mRNA long-read kit. So I think we are good. And again, we will come with another episode of Inside Takara Series. So we will announce the episode soon. So for now, I would like to thank all of you. Thanks, Ning. Thank you, all of you. Have a nice day. Take care.
Thank you, everyone.
Bye-bye.
Yeah, have a nice day. Bye.